Hello, my dear students. Today we begin an exciting journey into the digestive system. Have you ever wondered what happens to food after we eat it? How does it turn into energy and nutrients for our body? In this chapter, we will learn how different organs work together to break down food, absorb nutrients, and provide energy for life. Let's explore the amazing world inside us, the digestive system. Syllabus: the structure of a tooth, different types of teeth, structure of a tooth to be discussed with the help of diagram, functions of different types of teeth and dental formula for an adult. Digestive system: organs, digestive glands and their function including enzymes and their functions in digestion, absorption and assimilation of digested food. Finally, we should know organs and glands of the digestive system and their functions with reference to digestion, absorption and assimilation as a brief idea of peristalsis. This is the image showing the digestive system of human. Why the digestive system is needed or required? Food cannot be directly used by the body. It must be broken into a soluble, absorbable form to be absorbed by the blood. Some foods like cane sugar dissolve easily in water, but most foods must be broken down into smaller units so they can pass through cell membranes of the gut wall. Definitions or digestion: breakdown of naturally occurring foodstuffs into diffusible form, or any change making food soluble and absorbable through living membranes. Enzymes, they are very basically important because they play a major role in digestion. The general characteristics of enzymes are following. They are protein in nature. They are destroyed by heating. They act on one kind of substrate which is specific. It always form the same end product. Only affect rate of chemical reaction, don't alter reaction itself. It acts like catalyst, used again and again, work only at a particular pH, acidic or alkaline. It is active within a narrow temperature range, that is 35 to 40 degrees Celsius, which needs optimum temperature. The digestive system: the components are alimentary canal and digestive glands. The alimentary canal is a muscular tube from mouth extended up to the anus. Its length approximately or nearly 9 meters. It is highly coiled structure, which especially in the area of small intestine. The different regions of the alimentary canal have different structure and function. Digestive glands: it present in alimentary canal lining. It is associated glands are liver, pancreas and 3 salivary glands uh, found in the buccal cavity or oral cavity. Organs of the digestive system: the mouth cavity, the space where food is chewed and mixed with saliva. Lips, function is closing mouth, sucking and sipping liquids, speaking, perceiving touch and heat. Tongue is a muscular structure which helps in manipulating food during chewing, tasting and cleaning food particles in the mouth. It also helps in speaking. The teeth or the dentition, the arrangement of teeth. Function: break food into smaller bits. Smaller teeth, relatively larger surface area for enzymes to work. Heterodont dentition: humans have different types of teeth: incisors, canine, premolar and molar. They are contrasted with homodont dentition. All types of teeth are same. Example: lizard, frog, etc. Types of teeth: adult human dentition, which have 32 teeth. Incisors are 4 front teeth in each jaw, broad and chisel shaped structure. Their function is cutting and biting food. But the canines are present one on each side of the incisors per jaw. It is sharp, conical and pointed. Its function is tearing food. Premolars: 2 on each side of each jaw, total 8 teeth. Each has 2 cusps, also called as bicuspids. Their function is grinding and crushing food. Last 3 teeth on each side in each jaw, total 12, show the larger surface area than premolar. Its function is principal grinder and crusher of food. The last molar in each jaw is called as the wisdom tooth, which appears at 17 to 20 years of age. Role of teeth: it helps in digestion by breaking food into smaller parts. It helps in speaking, pronunciation of sounds like thick, thin, the etc. It adds to facial beauty. Confusion over molar: premolars of a child are sometimes called molars. Their true molars are permanent, appear only once. Premolars appear in 2 sets. To avoid confusion, keep premolars and molars as separate categories. Mammalian teeth appear in 2 sets. which is called as diphyodont arrangement. Milk teeth are 20 teeth, appear by 7 to 8 months, and it is completed by 2 years. Permanent teeth or 32 teeth, it replaces the milk teeth gradually. Temporary teeth or milk teeth or deciduous teeth or permanent teeth. The temporary teeth, also called as milk teeth or deciduous teeth, appear at about 2 years of age. They fall out because roots get dissolved. They are replaced by permanent teeth around age of 12. Dental formula of human expresses the number of different teeth like incisors I, canine C, premolar PM, and molars M. For half of one jaw. In case of childs, approximately 2 years, there is no milk teeth, there is no premolar, no third molar, so they have 2120 in each jaw. In the each half of the each jaw, 2120, that means total 20 teeth. In case of adolescent, 17 to 20 years, permanent teeth before wisdom teeth, 2122, that means 28. And in case of adult after 20 years, includes wisdom teeth, they are 2123, means total 32 teeth. Structure of a teeth, general parts of a tooth: crown, which is exposed above gum; root, it is embedded within jawbone; and neck is the constricted part between crown and root. Vertical sectioning of the tooth shows enamel, it is the hardest substance, or it covers crown. Dentine, it forms bulk of teeth, harder than bone but softer than enamel. It contains tiny canals with cytoplasmic strands. Cement, it is a bone-like material cover covering root, fixes tooth in place. Pulp, it's a soft tissue inside pulp cavity. It contains blood capillaries, lymph vessels, nerves, are supplied tooth with nutrients and sensation. The salivary gland. Saliva is secreted by three pairs of salivary glands. Parotid gland, which is located just in front of and beneath each ear. The image showing the position of parotid, submandibular and sublingual gland. Submandibulars are lying close to inner side of the lower jaw on each side, and the sublingual glands are located below the tongue. The secretion and flow. Ducts from each gland transport saliva into the mouth. Small quantities are secreted continuously. Their flow increases during eating. Even sight, smell or taste of food can stimulate extra flow, leading to watering of the mouth. How much saliva is produced daily? About 1000 to 1500 ml per day, or 5 to 7 empty bottles of soft drink. Saliva composition. Saliva is slightly acidic fluid, pH nearly 6.8. It has 99% of water and salts, mucus and enzymes like salivary amylase or ptyalin. Functions of saliva: it moistens and lubricates inner lining of mouth cavity and surface of tongue. It helps in speaking and swallowing. It moistens and lubricates food, help in swallowing, act as solvent. It dissolves food particles. It stimulates taste bud. It helps food particles stick together into a bolus so they can be swallowed easily. Digest starch. The salivary amylase ptyalin maltase converts starch into maltose. It explains why boiled rice, even chewed well, tastes sweet. Cleans the mouth and destroys germs. It prevents tooth decay. Prevents dryness of mouth. Less saliva, it will produce thirst, and helps maintain water balance 1.17. Swallowing and peristalsis. Swallowing is a coordinated action involving tongue presses upward and back, forcing bolus into throat or pharynx. Soft palate, it closes nasal passage, prevents food entry into nose. Larynx or voice box, it moves upward. Epiglottis closes windpipe, it directs bolus into the esophagus. Peristalsis is a wave-like muscular contractions of circular muscles of the gut, pushes food along esophagus, stomach, then stomach and ultimately intestine. It occurs in all regions of gut. Slimy mucus secreted by gut walls lubricates food for easy passage. Protective reflex: if food accidentally enters the windpipe, body reflexively expels it by coughing and choking to clear airway. Swallowing and peristalsis, part A, showing the breathing. When larynx lowered, glottis open and air passes through windpipe. Part B, or swallowing, larynx raised, glottis closed by epiglottis, prevents food from entering windpipe, bolus directed into esophagus. Part C, or peristalsis, it shows wave of constriction, that is muscles contracting, and wave of relaxation pushing bolus downward through esophagus. Esophagus structure a muscular tube carrying the food throat to stomach. It passes through diaphragm close to the backbone. There is no digestive enzymes and secreted here. Only function is to transport the food. Movement, food movement is occurred by peristalsis, wave-like muscular contractions of the esophagus. Stomach, nature of stomach, it is a elastic bag below diaphragm. It's a capacity 2 to 3 liters in adult. Highly muscular walls, they churn food with gastric juice. Openings, pylorus or pyloric sphincter, gatekeeper between stomach and intestine. It prevents premature passage of food into duodenum. It prevents regurgitation from intestine. Cardiac sphincter, it is located on the front end or junction of esophagus. It prevents backflow of food into esophagus. Vomiting. It is triggered when stomach is overloaded or disturbed. Mechanism, cardiac sphincter opens, antiperistalsis or revert contraction pushed contents back up or expelled through mouth. Gastric juice secreted by inner lining of stomach. It is colorless, highly acidic, contains HCl, salts, water and mucus. It has enzyme like pepsin which digest protein. Its function is to kill harmful germs in the food. It provides acidic medium for pepsin activity. Pepsin formation, it is secreted as inactive pepsinogen. Then it is activated by HCl. It will form pepsin, which is the active form. It converts proteins into peptides. Renin, when it will form prorenin to renin, acts on milk protein or casein, it converts into soluble paracasein, which is a peptide. Food in the stomach, it stays for nearly 3 hours, mixed into semi-solid chyme. Pylorus opens intermittently, allow chyme to move into intestine in small amounts. Small intestine, its tube is nearly 7 meters long and 2.5 centimeter wide. It is coiled and folded in the abdomen. It is divided into 3 subregions: duodenum, ileum, jejunum and ileum. Duodenum is the short upper part next to the stomach. Name duodenum means 12 finger breadth. Its common bile duct opens here. Jejunum next 2 meters long. It means empty or often empty at depth. Ileum, it is about 4 meters long. Name ileum means twisting movement or peristalsis occur within this structure. Structure of ileum, inner lining has villi or finger-like projections, and its function is enormously increased surface area, which is 8 times longer than the outer body surface. Each villus is covered by single cell thick epithelium. It contains artery, vein, capillaries, a lacteal or lymph vessel. Its role is facilitate absorption of digested food. Adaptations of ileum, it is very long, so it has long or large surface area. Their villi and microvilli further increases absorption surface. Single cell epithelium, which is responsible for quick diffusion, and narrow and slow movement of this structure help in more absorption time. Digestive juices entering small intestine. Bile from liver stored into gallbladder. There is yellowish green watery fluid, contains bile salts, bilirubin or biliverdin. Its function is to neutralize acid chyme or sodium bicarbonate. It emulsifies fat. It breaks into tiny droplets for easier enzyme action. Pancreatic juice, it is secreted into the duodenum via pancreatic duct. It is rich in enzymes for carbohydrate, protein and fat digestion. Pancreatic juice, which is produced in the pancreas behind the stomach. It ducts opens into duodenum along with bile duct. Contain 3 enzymes: amylopsin for pancreatic amylase, starch and maltose will be destroyed there, and trypsin, which acts on protein and polypeptides, which ultimately form small peptide and amino acids, secreted as inactive trypsinogen activated by enterokinase from duodenum. Steapsin or lipase, it emulsifies fat and convert fat into fatty acids and glycerol. Summary of pancreatic enzyme: trypsinogen, it will form enterokinase, then trypsin, then peptide and ultimately amino acid. And amylase will form the starch and maltose. Steapsin will form emulsified fat into fatty acid and glycerol. Intestinal juice or Saccas entericus, it is secreted by the inner wall of small intestine. It contains several enzymes like eripsin or peptidase, which breaks peptides into amino acid. Maltase, which acts on maltose to form glucose. Lactase also acts on lactose to form glucose and galactose. Invertage or sucrase, it acts on sucrose, which form needed glucose and fructose. Lipase, it has fat, so it will convert fat to fatty acid into and glycerol. So the final products are protein থেকে amino acid. From the carbohydrates it will produce glucose, fructose and galactose, and from the fat it will produce fatty acid and glycerol. Absorption of food. Final products of digestion are absorbed in the ileum. Amino acids and simple sugars are absorbed into blood capillary via portal vein, and then it will go to liver and ultimately move into circulation. Fatty acid and glycerol, it is absorbed in the lacteals or lymph vessel in the villi. Lymph, then it will transport into blood. Food stays 4 hours in ileum before moving to large intestine. Large intestine is 1.5 meter long and 3 parts. Cecum, it has small blind pouch. At the junction of small and large intestine, it has vermiform appendix. It is vestigial. It can cause appendicitis. Colon, it is a broader tube, nearly 1 meter. It is divided into ascending, transverse, and descending colon. Rectum, it is the last part, last 15 centimeter. It has sphincter muscles which controls exit. No enzyme is secreted. Absorbs excess water and some salts. It converts contents or semi-solid feces stored in rectum expelled by defecation. So components, 75% water and 25% solid matter, that includes undigested food, bacteria, dead cells or salts. This is the summary of digestion in various parts of human alimentary canal. You can go through the table. Composition of fishes: nearly 30% dead bacteria, 10 to 20% fat, 2 to 3% protein, 30% roughage, which is the undigestible fiber. Bad odor due to bacterial action, and composition varies with diet. Assimilation of food. Assimilation means conversion of absorbed digested food into body material. After absorption in small intestine, sugar and amino acids absorb into blood, then liver via hepatic portal vein circulated to body, used for energy, growth and repair. Fats is absorbed into lymph vessel or lacteals carried by lymphatic system. Blood is used for enzymes, storage and remembrance or hormones. In simple words, whatever we get gets break down, absorbed and then used by our body to grow, repair and get energy. Carbohydrates or glucose. Extra glucose are stored as glycogen in the liver, like money in a savings account, but when energy is needed, glycogen will form the glucose and it will produce the energy. Functions of glucose: main form fuel for respiration is ATP, helps in making other important also. Protein or amino acid. Amino acids are building blocks for proteins. They are needed for muscle action, enzymatic action and hormonal action. They cannot be stored like glucose or fat. Essential amino acids are deamination in liver, nitrogen part, urea, it will form different excreted product, and remaining part, it is converted to glucose via gluconeogenesis and then backup energy. Fats or fatty acid and glycerol. It is absorbed into lymphatic system, then blood near heart. Its roles: used to build certain compounds in cells, stored as subcutaneous fat, under skin, keep us warm and good and around organs. Sources of energy when we curves run low. The liver is described as the largest gland of the body, nearly 1500 gram average weight, reddish brown, located in the upper right side of the abdomen and below the diaphragm. Functions of the liver: other than production of bile, they control blood sugar level, they control amino acid level, they are responsible for synthesis of RBC, production of clotting factors, production of anticoagulants, regulates blood volume, destroys old RBC, storage nutrients, excretion, they excrete toxic metabolic poisons, heat production, produces body heat via metabolism, detoxifies drug and alcohol. Certain experiments on digestion: the action of saliva on starch. Experimental setup: prepare starch suspension in water, divide into test tubes, test for starch with iodine, add saliva to 1 drop, keep warm, nearly 38 degrees Celsius. Then iodine test becomes negative, sugar test becomes positive, starch converted to sugar via tiling. Understood? Next, we will see the action of pepsin on egg white. Egg white or protein is boiled water and filtered. Suspension is prepared, then suspension divided into test tube A, B, C, D, E. Reagents added. A means pepsin only, B হচ্ছে HCl only, C হচ্ছে pepsin plus HCl, D হচ্ছে HCl plus boiled pepsin, and E means the board. E means pepsin plus sodium bicarbonate plus alkaline medium. All test tubes are kept at 35 degrees Celsius for 10 minutes. Observation: A, pepsin only, there is no change. B, HCl only, there is no change. But C, when pepsin and HCl was both, solution turns clear, protein digested. When it was D, then boiled pepsin and HCl, there is no charge, enzyme destroyed by heat. E, pepsin and alkaline medium, there is no charge, peptin is inactive in alkali. Conclusion: pepsin digests protein only in acidic medium, acid alone cannot digest protein, so heart destroys enzyme activity, boiled pepsin inactive, pepsin does not act in alkaline medium. It describes common lab test to identify different food compartments. Test for glucose is sugar, so its reagent is Felling's reagent. Method: add glucose solution, Felling's solution in a test tube, heat gently, and observation: blue to brick red precipitate, which is indicates the glucose is present here. Application also used to test sugar in urine in case of diabetes mellitus. Note: cane sugar needs to be hydrolyzed part, which is the dilute HCl and dilute NaHCO3. Test for starch. Method: add iodine solution to food sample. Observation: blue by black color, so starch will appear. Example: rice, potato, bread given positive test. Test for protein materials: hard boiled egg white. Method: add dilute nitric acid, heat, it will wash, then ammonium hydroxide will be produced. Observation is white to yellow to orange, so it is indicates that protein is present. Test for fat and oils. Method: rub groundnut, walnut or butter on paper, and observation: paper turns translucent. So test for water: cobalt chloride paper, then blue to pink when exposed to moisture, cannot be tested directly with cut potato, banana, etc. Test for minerals: burnt potato, banana in crucible, ash remains, then identify the minerals present in it. So digestion is aided by enzymes and organs working together. Teeth and saliva starts the process. The stomach digests protein, and the small intestine complete digestion and absorption nutrients with the help of bile and pancreatic juice. The colon absorbs water, the rectum stores waste, and the liver produces bile and perform vital functions. Food tests like the iodine test and filling test help detect nutrients. So dear students, today we explore how the digestive system works by a well organized manner, a well organized machine, from chewing food in mouth to breaking it down with enzymes and juices and finally absorb nutrients in the small intestine. Every organ from the stomach to the liver and pancreas plays a crucial role in ensuring our body gets the energy and metabolism block it needs. Truly, the digestion is the bridge between the food we eat and the life we live. Thank you for watching. Keep learning, keep curious, and I will see you in the next class.